Good afternoon. Thanks so much for joining us on this incredible day. It's an incredible day because it's part of our 30th anniversary celebration of the Beckman Institute, and we're delighted to have amongst us some of the Beckman Foundation members who have traveled from afar. Some of them, the gentleman in the front row, Larry Klein, from quite afar, not only from San Diego, where he's located, but just before that, literally days before that, all the way from East Africa. So I say all that because actually this is one way I can introduce Arnold Beckman and the significant impact that he had. We've all heard in many ways that story, but it's the presence of the Beckman Foundation coming from literally thousands of miles away, recognizing that this is what Arnold would have wanted them to do. And I think that speaks really loudly to the commitment that the foundation all has. And so I really thank you for being here and contributing to our 30th anniversary by your presence, which is so critical. The Beckman-Brown Lecture was established through funds from the Beckman Foundation in honor of both Arnold Beckman and our founding director, Ted Brown. Both of these are, I think, true pioneers in the trails that they blazed, and then they came together to blaze yet another trail, which is the establishment of this incredible institute. And so we are, as I mentioned, celebrating our 30th anniversary, and as part of that, I have already mentioned the Beckman-Brown Lecture, but I will call your attention to two other events that will take place immediately after. And so please continue to stay around and join us. When the lecture ends, there'll be some time for refreshments. There'll be in the atrium, you can mingle and ask our speaker or chat amongst yourselves any of the questions that you might have and not get answered and want to discuss them further. And then at 5:30, there will be some snippets of research stories, mind you, that will be delivered, short stories, by our graduate students. And they're eager to tell you, and postdocs, quite eager to tell you about their own research. To introduce the Beckman-Brown Lecture, it's really a great honor to have our founding director, Ted Brown, here. Before I call him up to the stage to introduce Yaakov Stern, who is our third Beckman-Brown Lecture, I'd just like to qualify everything that is coming with the following remarks. As I said, he's our third Beckman-Brown Lecturer, and our first Beckman-Brown Lecturer, Francis Arnold, went on the following year to receive the Nobel Prize in Chemistry. So, next week happens to be Nobel Prize week, in case you haven't paid much attention. I'll be watching very closely for the Nobel Prize in Medicine and Biology, Medicine and Physiology, right? Yeah, so there, the bar has been set. My mother likes me. It's really a great, so we select the Beckman-Brown Lecture with input from our faculty here, and there was no question that Yaakov was going to be the selection, but I did run it by Ted Brown, and he got the first Beckman-Brown Lecture spot on with Francis Arnold. And so I just said I have to keep going back to Ted to vet this and make sure it's the right choice. And indeed, he gave that overwhelming endorsement. So Ted, please come up and introduce Yaakov.
Thank you, Jeff. Thanks. One of these mics I should use here. Thank you, Jeff. It's always such a thrill for me to come back to the Beckman Institute, and especially to come to this room and talk and see all those faces out there and realize that we've come a long way in 30 years. I'm happy to introduce our Beckman-Brown Lecturer this year, Professor Yaakov Stern. She's from the Columbia University College of Physicians and Surgeons. And I thought it'd be interesting to note that we're celebrating our 30th anniversary, and 30 years ago, not quite this time of year, but in the spring, we had the inaugural lecture opening the Beckman Institute held in the Follinger Auditorium. And that time, the lecturer was Gerald Edelman, who was at that time at the Rockefeller Institute for Medical Research. And some of you maybe were there at that time. Edelman talked that time about his model for brain functioning, which he called neural Darwinism. So we have an interesting continuity of neurosciences here, and I'm sure that as the years progress, we'll have even more. Dr. Stern has devoted his career to studying an important topic, what happens to our cognition as we age? In particular, what's Alzheimer's disease, and what can we do to slow it down? Or even someday, perhaps prevent it. Professor Stern's work includes large-scale imaging studies to identify the unique neural networks underlying the major cognitive capacities affected by aging. One long-term study has been devoted to modeling the natural history of Alzheimer's disease. He's developed a model which he refers to as cognitive aging, and he will be telling us about that today, among other things. So, Dr. Stern, it's yours.
Well, it's a pleasure to be here. I don't expect a Nobel Prize. I'm certainly not. Let me just see. Good. So it's really an honor to be here to give the Beckman-Brown Lecture. I'm honored to be the third lecturer, and I'm really very much honored to be here on the 30th anniversary of the Beckman. It's a wonderful institution. I know many people who work here, so it's just a pleasure. Today I'll talk to you about this concept of cognitive reserve. It's a theory, it's an idea, and it really is an evolving concept. And towards the end, I'll explain to you that it's really evolving faster than I could imagine. But before I get into the concept of cognitive reserve, I thought I'd just introduce the reason that we think about it. So there's two concepts to talk about. The first one is cognitive aging. So this is a graph of age from 20 to 80, and this is the higher the score, the better. And you can see here's this one line where things stay the same or probably get better with aging. That's things like vocabulary, crystallized knowledge, knowledge of words, and things like that. But you can see these other lines, which represent reasoning, memory, processing speed, tend to go down with aging. This is cross-sectional data. That is, different people were tested at the different age groups. It's not longitudinal. And you notice that it tends to get a little faster later in age. But cognitive aging doesn't just begin at age 60 or 70. There is some loss earlier too. But there's a lot of individual variability as well, as what we'll talk about. So that's cognitive aging. Our understanding of what causes cognitive aging is not great. We know that there's a whole set of measures in the brain that we can see decline with aging. So this is the volume of the brain. This is the thickness of the cerebral cortex. The white matter is what protects the axons and allows signals to be transferred more rapidly. And the integrity of that white matter goes down with aging. And there's these things called white matter hyperintensities that you can see on scans. They're thought to be microvascular changes. They get worse with aging. So somehow our thought is that part of the cause of cognitive aging is these brain changes, although we're still trying to figure that out. And then there's Alzheimer's disease. Alzheimer's disease is different than normal aging. It's a disease. It's the most common form of dementia, which is a cognitive and a functional impairment. And the prevalence of this disease increases with age. Why is it so prevalent now? People are living longer because of better vascular care, better heart care, et cetera. People are living longer, and the prevalence tends to increase with age. What underlies Alzheimer's disease? Well, there's a lot of different pathologic changes. The ones you've probably heard most about are plaques and tangles. This is an amyloid plaque. This is this tau tangle. The amyloid plaque is not in the neuron, the tau tangles are. And there are two hallmarks of the disease. It used to be that you couldn't diagnose someone with Alzheimer's disease definitively until you could look at their brain post-mortem and see these plaques and tangles. Now we have PET scans that can actually measure the amount of amyloid in a person during life. So this is a very early paper utilizing that PET scan. Here's people with Alzheimer's disease where you see those bright colors. That's a lot of amyloid in the brain. Here's healthy elderly people with nothing. And then what was interesting, this was one of the first papers that said, look, here's some healthy elderly people cognitively intact, but they already have amyloid burden. And it turns out that if you take people over age 60, maybe a third of people have significant amyloid burden, although they don't have Alzheimer's disease, they don't have any cognitive problem. And then there's tangles. Now we can image tangles too. This is much more recent. And sort of the same story. Here's someone with a sample of a brain with no tangles at all, and you can see that it begins over here, it spreads, it gets worse and very worse, and as the tangles get worse, cognition typically follows. So now we can actually measure these two Alzheimer's disease-related pathologies in people. And the reason I went through all of that is to stress the idea that what we typically think of is that cognitive outcomes or functional outcomes are related to brain change or brain damage. So Alzheimer's disease should be a function of the plaques and tangles. Age-related changes should be in relation to these kinds of brain changes that I pointed out to you. And the concept of reserve came forward because there's not a one-to-one relationship between the measures of brain and their outcomes. I'll show you later that two people could have the same amount of brain damage and one person can look much better than the other. So this concept of reserve was introduced to explain that disjunction between the two. That's the simplest way to explain it. This is a review paper that we wrote recently where we got 32 people together to explain, to come to consensus on these various terms that I'm going to talk about today. This sort of an introduction. The concept of reserve now is something that's well studied. Many people have different ways of describing these phenomena, so this was an attempt to do it. So a lot of the definitions I'll use come out of this white paper. The first concept that came out is a concept of brain reserve. I didn't make this one up, but it figures into the story. The first person to use the term was Robert Katzman. Katzman and Terry were very influential investigators in the field of Alzheimer's disease. They used to be north of us in the Bronx. That's north of where we are at the tip of Manhattan. Later, they moved to California. So he was doing a longitudinal study of aging in the Bronx, and they were getting autopsies on all of the people in their study. And they described in this paper 10 women who had a lot of amyloid in their brains, but were cognitively normal right before death. And they were perplexed by this because typically the more amyloid, the worse the cognition, but these people seemed to be fine. And then they noted that they had very large brains. They had much larger brains than average. So they speculated that they started with these larger brains, had more large neurons, and this might be said to have a greater reserve. Now, what that word reserve is, we could talk about, but what they meant is these people had more neurobiological capital to lose. So somehow, because they had bigger brains, more neurons, they could lose more before it was affected. And this was the term brain reserve. And that term is still used. It's a little bit controversial. We could talk about it during the discussion. But that was the first term that was used of this nature. And the way I see brain reserve is like this. you have this brain reserve capacity, the amount of neurons or synapses that you have. One person could have more brain reserve capacity than the other. And given a lesion of a specific size, the person with the more brain reserve might not fall below this threshold, whereas this person with less reserve falls below this threshold and already shows some functional impairment. And I call this a passive model. The reason I say it's passive is that we don't talk about what the brain is doing to compensate for this damage. Basically, once you have enough damage to fall below this threshold, you start to have cognitive problems. And so that's the brain reserve concept. It's a passive type of reserve. We had a postdoc many years ago from Australia, Peter Schofield. He's back. He's a full professor in Australia. He was very fascinated by this idea. We were doing this big study, we still are, of aging in the area around the hospital in Washington Heights, that's North Manhattan, and he convinced the research assistants to measure the head circumference of all of the people in the study, and this is what he found. So this is head circumference from smallest to largest, and this is the percent of people with Alzheimer's disease, the prevalence, and you can see this trend where the larger the head size, the lower the prevalence of the disease. So it's a simple concept, but it's a powerful one. Yeah, it's just how big your head is. Yeah. There you go. Now, I'm jumping ahead in history, but because this is sort of a complementary concept, this concept was not introduced till recently in 2012. By Nyberg and colleagues. They're these people in the Netherlands in Scandinavia, that do mainly aging research on people with normal aging. And they said that that the real reason why some people age more successfully than others is that some people don't develop those age-related changes that I showed you. They don't show volume loss. They don't show white matter changes. So the relative lack of brain changes in pathology is the biggest contributor to the heterogeneity of cognitive aging. So it's a very important concept because I'm going to talk to you about the reserve against the pathology. But the other idea, which I think is a very important one, is that some people don't develop these brain changes. And there are lifestyle factors, the same lifestyle factors that I'll talk to you about that contribute to cognitive reserve, that appear to contribute to brain maintenance as well. So brain maintenance, the definition we gave in that white paper, individual differences in the manifestation of age-related brain changes in pathology allow some people to show little or no age-related cognitive decline. So that's really a very important concept that I think is quite well accepted. And here's an example of brain maintenance. This is an old paper by Michael Valenzuela. He measured hippocampal size. Hippocampus is an area in the brain that's important for memory. And he divided people based on this life experience questionnaire. It's a questionnaire that asks a lot of questions about cognitively enriching activities throughout the lifespan. And he found that the people with the higher life experience scores showed less of a loss of hippocampal size over 3 years than the people with the lower LEQ. So this is really suggesting that maybe there are some experiences that allow us to maintain our brain, to maintain brain size and brain integrity. Now comes cognitive reserve. And I'll just explain this to you before I get into the story of how the concept came about. In the idea of cognitive reserve is different than brain reserve in this way. Now we have two people with the same amount of brain capacity, but now this person could tolerate a bigger lesion than this person before he or she starts to have a problem. Why is that? Because there's some active compensation, something about the remaining brain that's allowing this person to tolerate a larger lesion than this one before they show a deficit. So I call that an active model. And what I'll do is walk you through some of the early evidence for this idea. Okay, so this is a schematic that Katzman put forth on advancing Alzheimer's pathology. So it's important to note this, that there are certain, perhaps, genetic factors that help initiate Alzheimer's pathology and other things that might promote it. But the real point is that the changes that are associated with Alzheimer's disease can begin up to 20, 25 years before a person starts to manifest a problem. They develop slowly over time. At some point, clinical symptoms appear. Later on, they could be severe enough to be diagnosed, and then they get worse over time. So the cognitive reserve idea would be that some people could tolerate more of this pathology before these clinical symptoms appear, and some people can tolerate less. So how do you go about studying this? So the way that I first was exposed to this concept was I was at a meeting, and there was just, before there was Twitter and all of that, you would go to meetings and just hear talk. And Katzman, people say, well, Katzman says that he finds a lower prevalence of Alzheimer's disease in people with higher education. That in general, when he's looked at certain populations in China here and there, and if people have higher education, they're less likely to have Alzheimer's disease. And I'm a neuropsychologist. I sort of thought that was a mistake because they're diagnosing Alzheimer's disease with a mental status test. We know that people with higher education do better on these tests, and probably it's just a diagnostic error. So the way to really look at that is not to just look at people who do and do not have Alzheimer's, but to start with people who are cognitively normal and follow them over time. It's called an incidence study. Follow people over time and look to see if there's differences in the percentage of people who develop Alzheimer's disease as a function of something that imparts reserve, in this case, education. So this is an early study, 1994. We had the ability to do this because we were in Washington Heights, where we were following these elders. So it's about 600 people we followed. They were cognitively normal, fine at baseline. Low education was cut at 8 years. And we found that the people with lower education were twice as likely to develop Alzheimer's disease over this period of time. Similarly, for occupational attainment, the people with lower occupational attainment were twice as likely to develop Alzheimer's disease. So what was our interpretation of this? I did not think that these education and occupational attainment cause people to not have Alzheimer's pathology. I figured that the Alzheimer's pathology was happening no matter what. So what we concluded was probably these people with higher education and higher occupational attainment could cope with the pathology longer before they got demented. And I don't know if I use the term, I use some form, but I could say it because Katzman had used this term, brain reserve. I said, this is not brain reserve, this is cognitive reserve. So that's where the term came from. It might not have been the best choice of words. We had a very interesting talk today about metaphors, and reserve has its own connotations if you just look it up in the dictionary, but that's how the term emerged. It's hard for you to see this, but just to show you, this is a meta-analysis in 2005, a lot more. These are studies that looked at education, occupational attainment, premorbid IQ, and leisure activities late in life. And anywhere where this bar doesn't cross this line, they found a protective effect. The same thing that I found. They replicated what I found. So here's the first initial study, 2 negative studies. Here's my study here. And then many, many studies have replicated this idea that educational attainment, occupational attainment, leisure activity are protective in the face of Alzheimer's disease. I have to say that I was telling someone today, I remember presenting my findings as a poster at the Academy of Neurology. I'm sure most of you are familiar. You just stand in front of and I had friends coming over and chuckling at me. Part of it was because there had been mixed results, but part of it was because the idea that something like educational attainment could forestall this bad disease people just didn't buy it. It just didn't sound right. I think now we're sort of used to it. Everyone knows this term, use it or lose it. It's sort of commonly, but it just was not a popular idea at that time. Okay. Then a little bit later in time, Jennifer Manley, who was my postdoc at this time, said, well, what about people who don't have Alzheimer's disease, just a normal aging? So we were following in the same cohort of people in Washington Heights, we were following people who did not have Alzheimer's disease. And what Jennifer noted was she looked at people with high versus low literacy, which is a better measure of quality of education than just years. And she found that the people with high literacy showed less of a decline in memory over time than the people with lower literacy. So this was applying this cognitive reserve idea now to not just Alzheimer's disease, but cognitive aging. I have to say that we found it in other analyses. It's not always found. This difference in slope as a function of education or occupation is not always found. And then the final thing that we did epidemiologically is, well, what about the people with Alzheimer's disease who they already have Alzheimer's disease? Does cognitive reserve help them have a better course? And that was a sort of a counterintuitive finding that we had. So I have this graph to show you what our reasoning was. So here I have Alzheimer's changes getting worse and worse. Here I have memory test scores, the higher the better. So you could have a person with high reserve and low reserve. Maybe the person with high reserve is doing a little better on the test. If you believe what I've told you so far, you could see that this change point for when the pathology begins to affect performance is later in the people with higher reserve than lower reserve. As a matter, so they can't compensate as well, and they begin to change earlier. The person with, this is the sort of the cut point where people are diagnosed. So the people with Alzheimer's with high reserve are diagnosed later than the people with lower reserve. And this point here is this final common endpoint. We posited that because if someone does have Alzheimer's disease, it's a horrible disease. No matter how brilliant you are, it really just saps all function. So if you put all that together, what you conclude is that once someone begins to show cognitive changes, the people with higher reserve should have a faster decline. And that's exactly what we found. This is the very first, this was in '99, very first publication of this. These are people with higher, these are Alzheimer's patients matched for severity at baseline. This is a memory test score. And we found that the people with lower reserve showed less rapid decline than the people with higher. This has been replicated over and over and over again. If it seems counterintuitive to you, just think about it this way. They've tolerated this pathology for so long till it's gotten very bad. So by the time they succumb, they sort of don't have much left and they crump. And we see this clinically very often. Very brilliant person, we finally diagnose it and then they'll show a very fast decline. So those were the three pieces of epidemiologic evidence that we had early on that got me very interested and started to convince me of this concept of cognitive reserve. Now, this is what I was thinking about then, and I have this graph to help explain it. So if you could have two patients with Alzheimer's disease and they were matched very carefully for disease pathology, for disease severity, their cognitive scores and their functional scores, one with high education and one with low, if you believe everything I'm saying, if you looked into their brains, what you would find is that the person with higher education had more advanced pathology. So let me show you on the graph, just in case that's not clear to you. So here I have Alzheimer's pathology getting worse. Here I have the clinical severity from control to mild impairment to mild AD to severe AD. So when the pathology is mild, you might have this situation where the person with high reserve is not demented and the person with lower reserve is already demented. Here there's more severe pathology. Given any amount of pathology, the person with lower reserve is going to be more affected than the person with high. If you draw a line this way, now you're matching for severity. They're both mild. This person has less pathology than this. So I was dying to try to test this idea, but at that point, we didn't have the autopsy data to do that kind of study. Now there are. 10, 15 years after that, there were. But we were doing studies of patients with Alzheimer's disease with an imaging approach, was with xenon. It's basically a blood flow approach. And we knew that this blood flow changes in Alzheimer's disease in a very specific way as a function of the underlying pathology. Today, there's a term for it that we didn't have then. Now, if you're up with the latest terminology, they call this the neurodegeneration. So the idea would be, if we could match for disease severity, would people with higher education have more of this neurodegeneration? And here's the results of this very early study. This was really the first study I ever published on cognitive reserve. So this is here 20, 20, and 20 patients, very carefully matched for disease severity. And if you see at this detector here, as the education gets higher, but below high school graduate above, there's more and more flow loss, suggesting more and more pathology, as if the people with higher education, even though they look to be the same severity, are walking around with more pathology. So I was very excited by this finding. I thought it was going to be on the cover of Science. It was not. But my friends can tell you that I never give a talk about cognitive reserve without showing this slide. It was just one of those, because what happened was I had the idea, we were doing the study, and we could just look at once we had the concept, the next day we could look at this data, and it just came out that way. So it's very exciting. And this has been replicated over and over again in various ways. Okay, so, and the other reason I like this slide is when you think about cognitive reserve, you need to think about three things together. And this is the important part. You need to think about the brain change or pathology. You need to think about the clinical outcome. So the brain change here is this underlying AD pathology. The clinical outcome here is mini-mental and function. And then the moderating effect, what is helping with the reserve? Here is education. We did the same thing with occupational attainment and leisure. So you need all of those to really think about the reserve concept. Okay, at that point, I was pretty convinced about the cognitive reserve concept, and I thought that if I could do functional imaging, I could really understand how it's, quote, implemented in the brain. And so this is sort of an idea of what we're doing now. I'll just give you an idea. I'm not going to go into huge detail. So what we do is we have young and old people. We measure everything in their brain. A lot of the stuff I showed you in that first slide, the volume of their brain, the cortical thickness, white matter hyperintensity burden, white matter tract integrity, et cetera, amyloid burden. We also measure their cognition. And we assume that there's some relationship between these age-related changes in pathology and cognition, and then we have all of our proxies for cognitive reserve, and we assume that these proxies will moderate between the brain and cognition. So there's this disparity. The brain measures don't perfectly predict cognition, and education or occupation can help us explain that. Then we give people scans while they're doing tasks. One is a working memory task, one is an executive function task. The only thing I want you to get a feel for is our intuition is that if two people have the same amount of underlying brain changes, but one is doing better than the other on the tasks, on our cognitive tests, we could figure out some differences in what they're doing in their brain by looking at the fMRI, the cognitive activation during these tasks. So maybe the people with higher reserve are more efficient. They don't have to activate as much. Maybe they can compensate. There's a lot of ideas like that. So we've spent a lot of time. This slide summarizes about 15 years of our approach. And you see what we're talking about here is sort of the task-related activation, the activation that's occurring while they're performing a task. And then a new concept that we've come up with, it's not so new, but now I think we had a very good handle on it. It's a little different, so I'll just present that to you very quickly. So we said, the thought is that, I'll walk you through it. Cognitive reserve allows people to maintain function in multiple activities in cognitive domains in the face of brain pathology. So if you trust this incidence data that I showed you, if someone is forestalling the onset Alzheimer's disease is not just memory, it's naming and other language functions, it's day-to-day activities, abilities. So cognitive reserve, there might be some generic type of cognitive reserve that allows people to forestall all of that. So if a particular brain network subserves cognitive reserve, it should be active across tasks with varying processing demands. So what we were positing is that maybe there's some kind of a brain network, a set of brain areas that no matter what task people are doing, allows people to do better in the face of pathology. So here was our thought experiment. We wanted to identify a cognitive reserve related brain activity that we could see in 12 different tasks in the scanner, whose expression correlates with IQ, and we could forward apply it to other tasks and would correlate with IQ, and whose expression moderates the relationship between cortical thickness and task performance. So we wanted a network that can explain why some people, given a certain amount of thickness loss, can do better than others on a task. So to put it short, my colleague, Chris Habeck, really does this kind of thing. We had 12 different tasks that people were doing in the scanner, and he pulled out a set of brain areas, found a set of brain areas with a very nice statistical approach that was active in all 12 tasks and was more active with higher IQ. So if you had higher IQ, you expressed it more. And then we took that network and we looked at it in a totally different task, and you could see that as this is people expressing this network in a different task, and as this is a measure of IQ, as people express this network more, their IQ was higher. So it seemed to be sort of a generic network that was related to IQ. And then here, this is sort of the test. We had this cortical thickness I spoke to you about. We had a pattern of cortical thickness that was related to a reasoning task, actually a reasoning domain. And what we were able to show here, follow it for you, the red are people with higher expression of this network, the black are people with lower expression of this network. And if you follow it down, you notice that as the thickness gets lower, performance gets lower on this task, but the red group, as the thickness gets lower, is doing better than the black group. So this, according to our definition, utilization of this network is imparting reserve. Okay. So that's sort of the cognitive reserve story. We are working very hard in a lot of ways to understand it better. There's many other studies that utilize the concept. What's this good for? So how would interventions work? So I think we could think of two things. To the extent that brain maintenance is a valid concept, can we help people maintain their brain? Or can we impart reserve to allow people to tolerate brain changes better? That's sort of the hope. Part of the reason I'm hopeful that that's the case are data like this. This is Marcus Richards, who runs a very large cohort of people in England that have been followed since birth. So Marcus knows for every single person, their sort of socioeconomic status at birth. This is a very old study, so it uses father's occupation. Their sort of cognitive status or IQ at age 8, their education by age 26, their occupation by 43, and then a measure of a crystallized word knowledge at age 53. And part of this was the question, well, you could say, well, Yaakov, look, someone is born with a higher IQ. Of course, they get a better education, they get a better job, and of course, they'll just do better in the face of pathology. So what Marcus was able to show is that there is an independent contribution of each of these, the cognition at age 8, education by year 26, occupation by age 43, each contributed independently to someone's cognitive status at age 53. So every facet of life experience is contributing to reserve separately. And so it's not like you have a certain amount of reserve, it's more of a plastic thing. And so that raises the possibility of intervention, even past age 53, supplying people with experiences that could promote reserve. This is another nice summary paper. This is a summary of a lot of epidemiology. where the investigators looked at a set of risk factors that were associated with Alzheimer's disease. If you can't read them, I'll read them off: diabetes, hypertension, obesity. So these are sort of more vascular kinds of things: depression, physical inactivity, smoking, and low education. And they said together, half of the AD cases, Alzheimer's disease cases worldwide, in the USA, 2.9 million, are potentially attributable to the listed factors. So a 10 to 25% reduction in all 7 factors could potentially prevent as many as 1.1 to 3.0 million cases worldwide. So they're saying that we have these lifestyle factors, things that are treatable, that might actually help us forestall Alzheimer's. Now, at this point, we don't have any treatment for this disease, so any kind of a lifestyle intervention would be very welcome. Now, I'll just show you a few other suggestive factors. Now, I feel very funny talking about physical activity, since some of the seminal work, or most of the seminal work on physical activity and its relationship to cognition was done right here. But in this analysis, again from the Washington Heights study, 2 things were looked at: the Mediterranean diet and physical activity. And people who were physically active and adhered to this healthy diet had much less of a risk of developing Alzheimer's disease than people who didn't either. And so you could see that. So there's a eating sort of heart-healthy diet, brain-healthy diet, and engaging in physical activity are good for you. Cognitive interventions is one of the things that you would love to be able to do. Can we give someone some kind of training, give them some computer game to play? And I've been a little, and many people have been a little disappointed on that aim. And again, here there's many people who are doing amazing work here on these issues. So the most cognitive intervention studies show a very small effect. Very often people get better on what they're trained to do, but not on other things. It's not clear that it generalizes to their day-to-day function. How sustainable is it? And it's really not clear if it's related to the rate of age-related changes or Alzheimer's. So it's been a very tough go, I think. So these programs like POSIT and those, it's just not clear that they're really very helpful. I personally feel, and there's evidence to suggest that just remaining engaged, doing the things you like to do, remaining active is really the key issue. There's been some very interesting studies of post-retirement. Retirement is a hard thing to study because you could say, well, maybe someone retires because they had to. But people have done very beautiful studies because there's different countries around the world that have forced retirement at different ages. And it's quite interesting. In the lower SES levels, retirement is a very bad harbinger for decline after that, because these people, I think, just sort of stop doing things, and they show a real cognitive decline. People in the higher socioeconomic status seem to do okay after retirement. I think if you look at the people who've retired and remain active and go back and take courses and become engaged in many other things. So I think this cognitive engagement is more important. This is the biggest study of 2,800 people, 65 or over, where they did cognitive training on memory, reasoning, and speed training. I could say a lot, but basically what they found is that if someone was trained on memory, they did a little better on memory tests. If someone was trained on reasoning, they did a little better on reasoning tests, but there was no transfer from one test to another. If the authors of the study were here, they might debate me a little, but that's my overall take of that study. This is another approach that's becoming very popular. This is the finger study. The F is for Finland. Now it's very cool to have very nice acronyms for your studies. There's a United States version of it that's called the pointer, which also stands for something, I'm sure. But look at what they're doing. This is 2 years: nutrition, exercise, cognitive training, monitoring and management of metabolic and risk factors. So a very aggressive multimodal intervention. In relatively cognitively normal people at baseline. And what they found was a small effect. but significant, statistically significant, where everybody in the whole study seemed to improve on the cognitive testing, but the people who got all this good stuff improved a little more. So this is very popular, but again, the effect sizes are not so great. So what should a large-scale project to enhance cognitive reserve look like? You would have to start with a healthy elderly population, and then we'd really have to think about the intervention. Should it be focused to some cognitive domain? Should it be multimodal? How long should it last? You might say, well, you have this lifelong exposure to these stimulating activities. How long will it take to show some effect short term? We need long-term follow-up. And then I think the only kind of outcomes that would convince skeptics is if we could influence the rapidity of cognitive functional decline or reduce incident dementia. A study like this, studies like this are starting, massively hard to do, very expensive, but there we are. Okay, I just want to say this. Because this concept of reserve is very popular now, there's a lot of people doing the work, and the people at the National Institute on Aging notice that people are using different terminology, they're using different measures, so they put out a grant for what they called a collaboratory. I never made up that word, they called it that. And the goal is to bring together researchers from basic animal through human studies in order to reach a consensus on operational definitions and research frameworks. So they put out a request for applications, which I got the grant, and we started already. They suggested 3 annual workshops. We had the first workshop already. You can see it. It's on reserveandresilience.com, A&D. All the talks are online. And pretty much what we got to at this first workshop is there's a huge diversity in the types of terminology people use and the way people think about these concepts. So the idea is to have a process over time to people to come to consensus on how they talk about these terms, how they define them, how they research them. And the key thing also is to include animal researchers. So I hope I've convinced you that the epidemiologic evidence is supportive of this concept of reserve. I think it's important to say that this reserve is malleable and influenced by aspects of experience at every stage of life. That's an important concept. And I've talked about 2 forms of reserve: brain reserve, which is supported by this brain maintenance. The more maintained your brain is, the more brain reserve you have, and cognitive reserve, which is sort of a more active process. And what I didn't talk about today is that this concept of cognitive reserve is applicable to a wide range of conditions. I've talked about aging and about dementia, but it's been applied to multiple sclerosis, to head injury, to schizophrenia, et cetera. So a lot of people are using it. It's a very useful metric and concept in a lot of different conditions that affect the brain. Accepted imaging studies can help clarify the neural implementation of reserve. I didn't get into that, but I hope that's true. Accepted or consistent definitions are going to be helpful. That's what this collaboratory is. And I think the money part of it is influence reserve and brain maintenance may delay or reverse the effects of aging or brain pathology. So careful, focused research will help us maximize the chance for successfully intervening in these conditions. So it was such a pleasure to be invited to speak here, and thank you very much for your attention.
Thank you so much for that very powerful message, clearly presented. We do have time for a few questions if there are some from the audience. I'll start with Art.
Okay, I'll start behind you.
No, you, I'm sorry. You, for a moment, thought, okay, that looks like that gentleman there.
So, an argument, I'm going to make an argument that is to battle with the reserve, but it's an argument about the assessment.
Right.
And I'll tell you an anecdote. I was a neuropsychologist. We had a person come in. We gave them all the standard batteries of tests, memory included, and so forth. And we said, look, you're perfectly normal.
Right.
It turned out, because the reason he came in was he was a minister. And he had traditionally been able to prepare his sermons out, long sermons, and give them without notes just spontaneously. But then he had a stroke, and now he had to use notes. So the argument here, if you see where I'm going, is that in his case anyway, we didn't have a powerful enough measure up there, and so the line where you have him going flat would have been going, he would have been going down. You see the argument?
Yes. So the argument is that maybe for some people these tests are too easy, right? They're too easy, and so you don't see. So people have delved into this and tried very hard to make the tests much harder, to use IQ-adjusted norms. But I still have colleagues who really, my colleague Richard Mayu for many years, he said, is Yaakov, they're just hiding it, which is sort of, but if someone truly is functioning well and you give them the hardest tests you can give them, so that's this area of controversy. I have epidemiologist friends who who say, well, these tests are really not good at diagnosing anyway, right? Which might be, but then I think we could move the bar and start to talk about function and get to some level of dysfunction that we would be convinced is real, like problem dressing. And I think if you do that, you'll find the same phenomenon even to these more clearly measurable endpoints as well. But that's, those are the kinds of debates that people have in the field. So that's well posed.
You alluded to the imaging study. Well, where is that going in terms of evaluating neural function? Is the MRI the endpoint, or is PET scan promising, or is there new things we don't know?
Yeah, so typically we use the PET scans. Well, in my case, we're using PET scans to look at the presence of amyloid and tau. There's a lot of other things you can measure with PET scans. You can just measure general brain metabolism, but typically we see the PET scans, or I do, as a way of looking for the underlying brain changes that might cause cognitive problems. And then typically in human studies, to get a feeling for the mechanisms underlying the thinking, what happens when people do a task, we use functional measures like fMRI, or we use connectivity, or people use EEG measures, and those give us more of an insight into the underlying cognitive processes and what are the brain areas that are performing these are involved in performing these tasks, and how hard are they working? And that's what I try to play with in trying to understand reserve.
More questions?
Yeah.
Oh, please.
I would hear you talk more about your task and variant network. When would you think that's doing? What is that?
Yeah. So, I mean, when I do, it's when you do these things, you believe them, but you also have to say, is this real? For me, it's like the fact that I can identify a pattern of activation that is generic across many tasks, whose expression correlates with IQ, and that I can show moderates between brain and cognition, proves to me, at the least, that it is possible to identify a generic network. Is this the one? I'm not sure. But we can test it, because now that we have this, anybody who has fMRI data, we can apply it to them and see if it works in the same way in their data. And what's really exciting to me about it is that it's very hard to measure reserve. So the kind of measures that we have very often are the risk factors. How much education did they have? How much, what's their IQ? How many leisure activities did they engage in? The other measure, I didn't get into it today, is sort of an interesting one. They say, given this person's brain status, is this person doing better than expected or worse? That's another kind of a measure of reserve. But if this kind of a network is for real, then we really have a measure. Like, let's say we have some intervention that could increase cognitive reserve, and we could show that this expression of this network moves. So, I'm hopeful. There's other people that have other approaches to identifying these resting bull networks or other generic networks. But I think, and we need to replicate it in different ways. But I try to be very cautious about it. But I'm very intrigued by the fact that we were able to do it.
Yeah, I'm so sad that your assessment of the cognitive intervention work, and I agree, that it's not shown very much, but I'm wondering if maybe the problem is we're sticking too closely to looking at behavior, because maybe what we're going to look at is changes in brain function as a function of the same behavior.
Yeah.
And it's kind of hard because brain panels want to see changes in behavior before they invest in the neural measures. And we're doing it backwards.
This is like a very important question. When we do an intervention, what do we look for? So I was telling someone a very funny story. This is a Beckman study, because we did this Space Fortress task, which is very well known to most of the investigators here. And we did it as it was a video game that was created by scientists in the age where you had to go and put quarters in a machine to play, Space Invaders. And they created, and there was cognitive psychologists creating. So it's this complex video game. And I worked with this guy, Danny Gofair, who worked with Manny Donshin and a lot of the other people here with Art Kramer. And we actually did a Space Fortress intervention where we had people do this Space Fortress, and we saw a little bit of effect on an executive task. And Danny, in the inimitable fashion, said to me, Yaakov, they worked on this task for 36 sessions. This is a very demanding task. You're just not measuring how they're getting better, which was a very hopeful statement on his part. But I think that's where these kinds of, if we really trust these types of networks that we can measure, they might be a very much more direct measure of an intervention's effect. Yeah, that was a long-winded answer, but I got a Danny Gopher story in there. Yeah. Yes. All right. Yeah, because I know you're going to ask a hard question. Yeah. Yeah.
So I enjoyed hearing it all put together, Yaakov, because we've known each other for too many years and we've shown a lot of decline, both of us.
I'm standing straight.
One of the other big cross-campus institutes here at the University of Illinois is the Institute for Genomic Biology. And there's a lot of sophisticated work at multiple levels of description, from single SNPs to GWAS to on and on, proteomics and so forth. Genetics. I'm wondering, these are individual differences we can characterize in animal models and humans too, at every level of description, molecular level. Do they affect brain reserve, cognitive reserve, or both? Because I didn't see them in your slides.
The genetics?
Yeah, the genetics, proteomics. Yeah,
so we're just starting to delve into that. So they have these, you can develop, let's say, a risk score.
Polygenic risk.
Thank you. That was, I was searching for that. So you can develop these polygenic risk scores. It's a summary. So we could develop one, let's say, for education. There is a polygenic risk score for education. It's sort of weird. It's a set of genes that can predict how much education someone will get. Now, not everyone gets that amount because life impinges, but so, and then there's a polygenic risk score for IQ. So people have already used those in a reserve-like fashion for Alzheimer's disease and shown. But I think we could delve much more deeply into it. We have, in the patients we test, we have all of the genetics, we have all the material for the omics of, and for the, various immunologic stuff. And so we're hoping that we can sort of ease ourselves in. Those studies, in the end, will take, I think, collaboration across multiple groups to get the kind of numbers that you need. But I agree that that's a very powerful direction to go. Now, what's going to happen? Is it going to be, are there genes that are associated with people maintaining their brains better? I would guess yes. Are there genes associated with people taking advantage of, benefiting from cognition? It's sort of an interesting question. I would guess yes. But I guess in the end, I'm still thinking that what they really do during their life has to make a difference as well. So that's the, yes, yeah.
Okay, so what I'd like to do is suggest that we move the conversation to the atrium and continue there. But before we do that, let's thank Dr. Stern for really a fascinating talk.
Thank you. Thank you.